Hello children, let us now talk about different types of eggs and the classification based on the amount of yolk, and second is on the distribution of yolk. So on the basis of amount of yolk there are three types. On the basis of amount of yolk which is present in the egg, there are three types of eggs present. When very less yolk is present, then we call that egg as microlithial egg. Second is when the moderate amount is present, then we call it as mesolithial eggs. And the third category is when the sufficient amount of yolk is present, then we call it as macrolithial or polylithial eggs. Let us see the first one and the example of each one of them. Microlithial eggs, yolk is very less. It is seen in human eggs. Yolk content which is present, it is actually the nutritive tissue. So because nourishment is provided by the mother to the fetus by placenta, so less yolk content can be adjusted. And if no sufficient material is present in the egg, so there has to be some kind of supply from the maternal tissue to the fetus. So we have that less yolk can be adjusted in case of microlithial eggs, and the example is human eggs. Mesolithial means moderate amount of yolk which is there. This is seen in case of frogs. In case of frogs, because yolk content is not sufficient for the complete embryonic development, and that is why from the eggs of frog, the structure which hatches out is larva. So if there is larva form, it is an intermediate stage. Had there been sufficient nutrient, there would have been a complete development, but as nutrient there is not enough, an intermediate stage hatches out from the egg and it accumulates that nourishment and then gets into the next stage. So that is the young one, young organism is fully formed. So what happens is that the sufficient amount of nutrition is not provided by the yolk, and later on when it accumulates the nourishment, then it gets into the next stage, that is the young organism. So when it is fully formed, then it is not completely coming out fully formed from the parent's body. In case of macrolithial, sufficient amount of yolk is present, and this is seen in case of birds and reptiles. Because there is enough nourishment present, so what hatches out from the egg is complete animal. Further growth takes place later on, but complete body structure is formed, that is miniature form of the adult. So totally the amount of yolk is significant, because how much development can take place depending upon the amount, it is classified like this, depending upon the amount which is present. Microlithial, mesolithial and macrolithial, which is also called as polylithial egg, is present. So this is one category of classification. The other category of classification is on the basis of the distribution of the yolk. So if yolk is evenly distributed, then such type of eggs are called as homo or isolithial, when uniform distribution of yolk is there. And this is seen in case of again human eggs. So human eggs are microlithial, that is very less yolk is there, and homolithial, that is uniformly yolk is uniformly distributed in the cytoplasm part, that is what we call ooplasm. The second category on the basis of distribution of yolk is telolithial. So when the yolk is present on one side, then we call it as telolithial egg, and the example is hen's egg. If we cut hen's egg after boiling it, then egg shows the yolk content on one side, and such egg is called as telolithial, that is hens or birds. So you all must have seen this, on one side it gets accumulated. The third one is the centrolithial. As the name tells, is that yolk content is in the center, and it is seen in case of insects. So in telolithial, it is clearly understood yolk is on the one side here. Telo means also, you know, towards the end, you can say, or towards one end. And on the other side, there would have been the cytoplasm and the nucleus. So one side is the yolk and the other side is the cytoplasm and the nucleus. So the upper end here, if we see, is the nucleus. So it becomes the animal pole because of the presence of the nucleus here, and the other part, opposite part, is the vegetal pole, whereas in case of insects the yolk is centrally placed and the cytoplasm is towards the periphery. Here we take one more terms, that is cleidoic eggs. So basically it means self-sufficient eggs. Cleidoic eggs are self-sufficient eggs, and this means the egg has enough yolk content to supply nourishment for complete embryonic development, and such cleidoic eggs are found in reptiles and birds. So they are polylithetial, the yolk content is sufficient and they have a shell around them, that is a protective layer. So this egg is self-sufficient, that means it is going to protect the embryo as well as it is going to supply the complete nourishment till the development is done. And that is why from such eggs, the young ones, when they hatch, they resemble the adults. The only thing is that they are miniature and smaller in size. So this is how we classify the eggs, on the basis of how much yolk is present and how the yolk is distributed. So now we have understood the egg structure as well as types of egg, and now let us talk about how human sperm and egg can be compared, or how are they different from each other. Let us compare the two gametes, the human sperm and the ovum. So we are going to compare them on certain points. The first point on which we are going to compare the structure. So the structure of ovum is circular or round cell, whereas sperm is a flagellate structure and it is a haploid cell. Plus sperm has 4 different parts, that is the head, neck, middle piece and the tail, whereas in case of ovum it is just a spherical or circular structure. So structurally also the two gametes, they differ from each other. Now the second point is nucleus. In case of egg, the nucleus is swollen, bloated, and it is termed as bloated with the nucleoplasm, where nucleus is large and swollen. In case of sperm, the nucleus is condensed with no nucleoplasm, and that means during condensation, we have already talked about it, that when spermatid changes to sperm like structure, there are certain changes that take place. One change was condensation of nucleus due to the loss of watery content, that is the nucleoplasm and the acidic proteins. So that is how nucleus is condensed. So here in sperm the nucleus is condensed, and in ovum there is sufficient nucleoplasm. Third point of difference is mitochondria. In case of egg, the mitochondria are scattered in the ooplasm, that is the cytoplasm which is in the central part, it has scattered mitochondria. In case of sperm, the mitochondria are spiral, ribbon like, and they are called nebenchern. They are found only in the middle piece. Coming to another structure, that is centriole. So in case of egg, there is no centriole, whereas in case of sperm there are 2 distinct centrioles which are present in the middle piece, that is the proximal and the distal centriole. Sometimes we see one more centriole which is at the base of middle piece and which is called as ring centriole, but it is seen sometimes only. So now if you can recall again, I told you that this proximal centriole is responsible for spindle fiber formation for the first zygotic division, because when egg and sperm they fuse to form zygote, there is no centriole in the egg. So centriole which is coming from or through the sperm, that is the one which is going to help in the spindle formation during the first zygotic division. Then the fifth point is motility, means they are able to move or not, whether they are motile or not. Ovum is a non-motile gamete, that means it is not going to move, or it remains at its position, whereas in case of sperm they are motile gametes. Because of the tail present, they are able to swim in the liquid, and that is why they are called motile gametes. So structurally, organally wise and functionally, that is whether they can move or not, they differ from each other in so many respects. But the common thing is that they are all formed from the sex organs by the process of meiosis, and they are haploid cells, both of them, and they also help in sex reproduction. Now the next point on which they can be compared is cytoplasm. In case of egg, the eggs, they retain maximum cytoplasm. So here sufficient or maximum cytoplasm is present, and this is because of unequal distribution of cytoplasm, and this results into the formation of one polar body, and maximum cytoplasm retains in the egg. We have discussed about this in the previous session. In case of sperm, the cytoplasm is very less, and it remains only in the tail region, and that is why female gamete contributes maximum in zygote formation. Next point is about the membranes or the envelopes, which are the protective cover. In case of egg, there are 3 membranes which are present. So we have discussed about this point also in the previous session: vitelline membrane, zona pellucida, and corona radiata. In case of sperm, there is only one plasma membrane which covers the complete structure. So they differ structurally in so many respects. Plus one gamete is motile and the other one is non-motile or stationary. Both are produced by the same process of meiosis. They are haploid and they help in sexual reproduction. So when they fuse by the process of fertilization, both are going to bring equal number of chromosomes or the genetic material from the nucleus, and the egg brings some extra chromosome through cytoplasm, because cytoplasm has mitochondria, and in mitochondria are present certain genes, and that is called cytoplasmic inheritance. But the 2 gametes are ready, and next they are going to fuse, that is fertilization should take place. But before that, we have to understand one more process that is essential in reproductive cycle, that is menstrual cycle. So in next part we will take up menstrual cycle. Thank you.